Good morning to all. Welcome to the Diamond Group of Institutions, Neet and K-Cet online classes. Today's class is plant genetics. So, in the fifth chapter of Principles of Inheritance and Variations, plant genetics and animal genetics are there. So, in the plant genetics, we have discussed, and in that plant genetics, some of the previous important questions or previous concept oriented questions we have discussed. Those are first one. Capital R and capital Y genes are made lie very close to each other when capital Y, and smaller, and smaller y, genotypes are hybridized if F2 generation will show. See now, the question is they given as a two parents, that is capital R, and capital Y, smaller, and small y. Here the two contrasting characters are they given in the crossing of the two parents, that is we are going to represent as a dihybrid cross. It is prepared as a dihybrid cross. But in the dihybrid cross, based upon the dihybrid cross, they given as hybridized F2 generation will show as. So in a dihybrid cross, F2 generation will be going to shows, and here they given as a before the starting initially R and Y genes of the males very close to each other. So here if the genes are very close to each other on the same chromosomes, which are the genes are very close to each other on the same chromosome. You can imagine this is the chromosome and these are the two genes R and Y genes. The R and Y genes are very close to each other. Such type of the genes are called as linked genes. That's some method, and the process is also called as linkage. Here, these two genes are their presence very close to each other. Very close to each other. The two contrasting characters are given means we are called as a dihybrid cross. But here they mention as an R and Y genes of the males very close to each other. These two genes are present on the same chromosome. That is represented as a linkage. The question is a linkage. You know that at the time of linkage and recombination. So that is pH more than it will be done to discriminate that it was done the Drosophil experiment. The Drosophil experiments are discriminate and they are differentiated from the pea plants done by the Mendel's experiment. So why those are the Drosophil experiments are different than the Mendel's pea plants experiment? So in the Drosophil experiments, he was explained about the concept of linkage and recombination. But in a Mendel's law, there also was taken two contrasting data that shows as a dihybrid cross. The two genes are not present on the same chromosome; they are present on different chromosomes. So that's why they can show the segregation. But here, two genes are present on the same chromosome. So these two genes are called as a linkage. So the in between are two genes based upon the distance based upon the distance in between the two linked genes based upon the distance recombination is going to depend upon recombination is depend upon the distance between the two genes on the same chromosome. So now you see, so this is the linkage and recombination concept, linked genes. So options are we have to identify in a F2 generation which are the character which is going to suit. Option A, segregation in 9 is to 3 is to 3 is to 1 is represented as a dihybrid cross. Dihybrid cross. If this line was not given, means genes of R and Y they are present on the same chromosome. That is remain except this one remains we can consider as this dihybrid cross at the time segregation of 9 is to 3 is to 3 is to 1 will not affect this. Because here in these are dihybrid cross genes are there present on a different chromosomes. So easily separation will take place. But the linkage and recombination genes are present on the same chromosome. And option B, segregation is 3 is to 1. It belongs into the monohybrid cross, it's not related. And higher recombination types of parents. Now you want to know clearly the recombination is depend upon the distance between the two genes on the same chromosome. Here, these are the R and Y genes are present on the same chromosome. These genes are called as the linked genes. They are present very close to each other. So you know, here they are present as very close to each other. At the time of the linkage concept, linkage is more and recombination is less. If same genes are present on a chromosome, two genes are the capital R and Y, those are present and same chromosome. But the distance of the same genes is more at the time they are easily separated, they are easily segregated. So they are easily separation take place and recombination is more. So now here see genes are present as a very close to each other. At the time recombination is less. What are the offsprings are there found? Those are the offsprings are they are mostly similar into the parents. Means recombination is less. So variations will not exist. If recombination is more, means in between the two genes, distance is more, these are the genes will be known segregation, separation will occur easily. So whenever separation is take place easily, variations will take place and the new characters will be known introduced. So offsprings will be known to show us a variation. So now according to the concept of the linkage and recombination, higher number of parental types, higher number of recombinant types. So higher number of the parental types means they are belongs into the in between two genes distance is very less, so recombination is less. So here in the question only they are given as close, these genes are arranged very close to each other, so that's the reason these are the genes are they are not separated easily because the linkage is more. So what are the offsprings are they found, those offsprings are they can shows more number of parental types. So answer is C. So if they have shown the recombinant type means in between the two genes they should be present as a longer distance, means much distance should be. So next question, point mutation. Point mutation is mutation says as a changing of the gene sequence. Sudden change of the gene sequence it's called as a mutation. So these are the sudden change of the gene sequence by the changing of single nitrogen base or single nucleotide. It's called as a point mutation. So now they given as a option A, loss of a gene, change in a base of a gene, addition of a gene, and deletion of a gene. So addition of the genes and deletion of the genes with and loss of the genes. Those are all we are going to refer as mutations. So but in these are the mutations, several types are there. That is point mutations and frame shift mutations. So in these are the point mutation. Change the single nitrogen base, otherwise single nitrogen base pair of a gene is called as a point mutation. So remain all of the that is addition and deletion and last of the genes are they belongs into the other type of the genes. If the sequence is changed, the gene sequence will be changed by the changing of the more than one micro bases, more than one codon. So more complete the sequence of the organism of the gene. Those are all the different types of mutations. But out of that, point mutation means single nitrogen base pair or nitrogen base will be going to change. It's called as a point mutation. Answer. The next question. Out of a 7 characters in pea plant studied by Mendel, the number of flower based characters. Mendel he was studied 7 contrasting characters in a pea plant. Those are the 7 contrasting characters. Those are the 7 contrasting characters are stem, height, position of flower, and color of the flower, color of the seed, shape of the seed, and color of the pod, shape of the pod. So these are the 7 contrasting characters studied by the men. So out of the 7 contrasting characters, in the question they are asking how many characters are that belongs into the flower base. So out of the 7 contrasting characters, Mendel he was represented the flower based characters are 2, that is position of a flower. The position of the flower dominant and the recessive represented as dominant is a axial and recessive is a terminal. Color of the flower violet and white. So out of the 7 contrasting characters, 2 contrasting characters are represented as a flower character. So answer is the next question. Which of the following cannot be explained on the basis of Mendel's law of dominance? So Mendel proposed the law of dominance, law of segregation, and law of independent assortment. So these are the law of dominant. We can see into the monohydrous, law of segregation and the law of independent submixer. We are seeing into the dihydrous. So now, so based upon the Mendel's law of dominance, which are the character it's not suitable. Otherwise, which character Mendel was not included under the law of dominance. So now you see option A. Out of one pair of factors, one is dominant and other one is recessive. If you take into the any contrasting character, so these are the contrasting characters. We have to take as a pair. So the pair depends as if you are taken as a one character as a stem height of a plant. The stem height of the plant we are going to depend as one is a dominant, it's a tall, and other one is a recessive, is a dwarf. So these are the two factors. See now two alleles will be found present in a one pair of character. The two alleles are one of the allele or one of the character written as a tallness and another one is a dwarfness. So out of one pair of factors, one is a dominant and another one is a recessive. Yes, this is a pair of factors. One is a dominant tallness and another is a dwarfness. It shows as a Mendel's law of dominance. Alleles does not show any blending and both of the characters recover as such in F2 generation. Alleles does not show any blending and both of the characters recover as such in F2 generation. This option D statement and set represent as a law of segregation. So in the law of segregation, alleles does not show any blending. After the completion of the F1 generation, we are going to depend in the law of segregation. So these are the alleles that does not show any blending. Will be get into the characters of both parents in a F2 generations. That is says as a law of segregation. But they are asking now which are the characters not explain the law of dominance. So law of dominance is different, law of segregation is different, but both are explained by Mendel only. Question asking from the law of dominance only. And option C, factors occurs in pair. Yes, factors occurs in a pair. That is a factor of law of dominance. And discrete unit controlling particular character is called as a factors. So controlling factors or controlling particular character we are going to depend as a factor. So option A, C, D they show the law of dominance and option B is a law of segregation. So out of the given out of the given four options, law of segregation belongs into D. So answer is D only. Next question. Fruit fly Drosophila melanogaster was found to be very suitable for experimental verification of chromosome theory inheritance by Morgan and his colleagues. See now, chromosomal theory of inheritance was proposed by Sutton and Bobel. And about this chromosomal theory of inheritance for the experimental proof, TH more than he was done the experiment of a fruit fly or Drosophila melanogaster. So, Drosophila melanogaster is a suitable example for the chromosomal theory of inheritance and also the linkage and the recombination. He was selected for these two experiments as a Drosophila melanogaster. So, based upon the which characters we can consider the page modern was selected for the Drosophila melanogaster. Option A, it reproduce parthenogenetically. Drosophila melanogaster can reproduce sexually, not as a parthenogenital. Parthenogenesis says as formation of new individuals without fertilization. Here, Drosophila melanogaster does not perform the parthenogenesis; it will perform production of offspring through the sexual reproduction. And option B, smaller female is easily distinguishable from the large male. See now here, Drosophila melanogaster, male and female flies are different in shapes. So larger should be present as a female and the smaller should consider as a male. But here they given as a smaller is a female, larger as a male. It is a reverse. Larger is a female, large in size, female fly, and the small size is called as a male. And a single mating produce two end flies. So for the single mating of these Drosophila melanogaster, many number of the offsprings or many number of the daughter flies will be going to produce. And option D, it complete life cycle in about 2 weeks. Yes, he was taken as a Drosophila melanogaster for his experiments. The life cycle of this Drosophila melanogaster is very less, that is at 2 weeks. And these are the male and female Drosophila flies are distinguishable, and they are produce many number of the offspring for the single mating, and they are easily survive on a synthetic media. So that's the reason Drosophila melanogaster was taken for the experiment by H. Moran. So it complete life cycle about 2 weeks. This is the correct statement. Because of the completion of less life cycle of the Drosophila melanogaster, H. Moran was chosen. H. Moran was chosen for his experiments. And next question. In Antherinum, two plants with pink flowers were hybridized. The F1 plants produce red, pink, and white flowers in the ratio of 1 is to 2 is to 1. What would be the genotype of the parent? Here. So you want to know clearly, Antherinum is the best example of incomplete dominance. Incomplete dominance means here by crossing of the two parents, two homozygous parents, that is red flower and the crossing of white flower in the F1 generation will be getting to the intermediate character. The intermediate character is pink flower. So now this pink is called as hybridized. See now in Antherinum two plants with pink flowers. Now these are the two plants. That is chapter or smaller. Two plants with the pink flowers are hybridized. We have done the hybridization or self-pollination. The F1 plants produce red, pink, white flowers. The ratio is 1 is to 2 is to 1. What would be the genotype of? So here the genotype of the incomplete dominance producing flowers in the F1 generation is capital R and small r. So the two characters R will not express, intermediate characters will be going to form in the F1 generation. So the ratio it should be present as 1 is to 2 is to 1. If you are taking as a two pink flowers, the pink flowers are will be bounded by the crossing of the two homozygous plants. One is a dominant and another one is a recessive. That is one is a red flower and another one is a white flower. If you are cross the two parents, you will get as an intermediate character as a pink flower. The two pink flowers are we are under hybridization. We should be get as a red, pink, and white flower. The ratio will be 1 is to 2 is to 1. Once it is. If you are going to done the crossing of these two.
One generation, that is capital A, capital A, capital A smaller, capital R smaller, and smaller smaller. So here they given as a hint, capital R is a red, smaller is a white. So now capital R smaller, these are the pink flowers. So total 4 offsprings are there formed. Out of the 4 offsprings, one it is red, two are pink, and one is white. 1 is to 2 is to 1. So the ratio of these hybridized plants crossed, they will be get as a 1 is to 2 is to 1. So now the genotype of the hybridized plant is they are given as option A smaller, option B capital R smaller, option C capital R, and option C smaller. So that genotype of the hybridized parent it should be capital R and smaller. Answer is B. In the next question, which idea is depicted by cross in which of the F1 generation resembles both the parents? So here the question it is by the crossing of the two parents, the two parental characters will be donated in a F1 generation. That is called as option A Inheritance of one gene. Inheritance of one gene we are also called as monohybrid cross. In this monohybrid cross, in the F1 generation, will be get into the any one of the parent only, not into the two parents. In the F1 generation, will be get into the any one of the parental characters. That parental characters should be present as a dominant. One of the dominant parental characters only expressing a monohybrid cross. And the codominance. Codominance is a crossing of the two parents, the two parental characters in F1 generation will be known to data. So that's why these for these are the codominance better example as a ABO blood group. And these are the ABO blood group. When we cross into the two parental parents, will be get into the two parental characters in a offspring. So answer is B, codominance. And then what about the incomplete dominance and law of dominance? Incomplete dominance means will be the intermediate characters in F1 generation. Law of dominance, light as a monohybrid class only, dominantal parental characters only will be don't express. But the question it is, we have to get the both parental characters in a F1 generation. So that is called as a codominance. Answer B. Number of linkage group in Pisum sativum. Here, the easiest method to identification of the number of linkage groups in a given organism or given plant. So here the given organism and the given plant linkage groups we want to identify means you want to know the diploid chromosome number of the organism. If you are taken as a Pisum sativum, its diploid chromosome number is 14. Haploid chromosome number is 7. So number of the gametes are equals into the number of linkage groups. So number of the gametes or haploid chromosome number is equals to the number of linkage groups. If you are taken as a Pisum sativum, its a diploid chromosome number is 14 and the haploid chromosome number is 7. If you are taken as a rice plants, rice diploid chromosome number is 24, haploid chromosome number it is 12. So what are the number of haploid chromosome number is there or what is the number of gametes? It is equal into the linkage number of the organism or linkage group of the organism. See now here the Pisum sativum question they are asking, it's a diploid chromosome number it is a 14 and the haploid chromosome number or gamete chromosome number it is 17. Number of linkage groups in a Pisum sativum is 7. Next question. The smallest of genetic material is affected by mutation. The smallest genetic material is affected by mutation. Options are retron, cistron, mutant, and exon. So out of these, first you want to know means of mutation. Mutation means sudden change of the gene sequence is called as a mutation. So now here, first what are the words are they given in the options for those you want to know. Retron represents as a unit of recombination. A segment of the DNA is involved in a recombination process. Cistron, a segment of DNA involved synthesis of mRNA, or we are also called as a protein. A segment of the DNA is involving a synthesis of mRNA or protein. It's called as a cistron. Mutant, unit of mutation. Exon, coding sequence of a gene. So now they are asking a smallest genetic material is affected by the mutation. So now out of the given option, largest component is a cistron. Cistron means a group of the or it's a segment of the DNA. It is involved in a synthesis of mRNA and protein. So after the cistron, next largest, this is the cistron, and later will be get into the exon. Exon, it's represented as a coding sequence. So in these are the systems, many number of coding and non-coding sequence are present. So the coding sequence are we call as exon. After the exon, next will be get as a retron. Unit of recombination, a small piece of the DNA is involved into the recombination, and after the retron will be get as a mutant. A small N2 base or base pair, it is involved into the mutation process. So smallest genetic metal is affected by the mutant. Out of the given options, smallest genetic metal is a mutant only. It's a unit of mutation. It's a small single nitrogen base pair or nitrogen base. It's called as a mutant. So smallest genetic metal is. And question number 10. Mendel's law of independent assortment can be demonstrated by. Mendel's law of independent assortment can be demonstrated by. So here Mendel proposed by the law of dominance, law of federation, and law of independent assortment. So that law of independent assortment can be demonstrated by option A, they given as a test cross. So test cross we are going to use to determine the genotype of unknown parent. So test cross means we are going to use it to determine the genotype of unknown parent, and back cross. Back cross represents as, so to increase the parental number, so we have to cross with a hybrid, crossing with a hybrid with any one of parent. So if we can done the crossing of the hybrid plant out of the hybrid plant, so any one of the parent plant, we can cross these two will be get into the offspring. That offsprings will be don't increase the parental concentration, otherwise increase the parental number. And monohybrid cross, single contrasting character is involved into the crossing, it's called as monohybrid cross, and dihybrid cross. Two contrasting characters are involved in a crossing, it's called as dihybrid cross. So here Mendel independent assortments are we are seen into the dihybrid cross. Why we are chosen as a dihybrid cross only? So after the completion of F1 generation, in a dihybrid cross will be going to get into the four gametes. Those are the four gametes will be drawn to 50% chances having to pair into the 50% with the other gametes. So that's right. So these are the independent assortment. By the process of independent assortment only, in the F2 generation, 16 offsprings are formed. So that is formation of the 16 offsprings. These are the 16 offsprings. What are the alleles are they present? These are the alleles are they are having equal chances to pair into the other allele. So that is the call as a independent assortment. So now test cross, back cross, monohybrid cross, and dihybrid cross. Test cross, back cross, and monohybrid cross does not show into the independent assortments. And monohybrid cross it shows the law of dominance law and the law of dominance, and back cross to test cross we are going to use into the detecting of genotype of the unknown parent, and we are going to use into the increase the parental characters. So these are the three, and remaining as a dihybrid cross. The dihybrid cross only can shows into the law of independent assortment at the time of formation of F2 generation. The next question: pure line is connected with development of. Pure line is connected with development. First, you want to know the means of pure line. Pure lines means, so production of the offsprings. Those are the offsprings are they are formed by the continuously self pollination. So what are the offsprings are the plants are they formed. The plants will be don't developed by continuously self pollination. So by the performing of continuously self-pollination, what are the offsprings are there formed? In that offsprings, intermediate characters, otherwise other parental characters or variations will not show. So that is called as a pure lines. So these are the pure lines are that can form by continuously self-pollination. So here pure line is connected with the development of homozygosity, heterozygosity, homozygosity and self-assortment, heterozygosity and linkage. You want to know homozygous and heterozygous. In a pair of contrasting character or in a contrasting character, a pair of alleles will be going to present. So you are taken as a height of the plant stem, height of the plant. This is the contrasting character, and it contains as a two alleles. One is a tall and other one is a dwarf. So here, if the two alleles are there having same similar, that is called as a homozygosity. Means capital T we call as homozygosity. If the two alleles are there present in a different condition, we call as heterozygosity. So pure lines are connected with the development of continuously self-pollination, and that is options homozygosity, heterozygosity, homozygosity, and self-assortment, and heterozygosity and linkage. Heterozygosity will not get. So because here the two different alleles will be don't present means with all as hetero. In the alleles are will be don't present means with all as homo. And homozygosity with self assortment. Pure lines are always formed by the homozygosity with self assortment. We are also called as a self pollination. Answer is C. The next question. In a plant, red fruit is capital R is dominating over yellow fruit, small r. Tallness is dominating over shortness, and if plant with capital T, small t, genotype cross with the small t. So what will be going to get? See the question it is here. They given as a two plants. The two plants are capital T, small t, and small t. If you are going to done the pollination, or if you are going to crossing of these two parents, one and two, parent one, parent two. So gametes will be going to get as a capital T, capital R, small t, and small t. So now this is the these are the gametes. So we done the F1 generation, means we done the F1 generation with an made into the Punnett square for this. So otherwise directly we can show capital R, small r, capital T, capital R, small t. See now, capital R, capital T, small r, capital R, small t. Here, capital R is dominating on the small r, capital T is dominating over the small t. So capital R is a red fruit, capital T is a tallness. So now this plant is represented as a red with tall plant, red fruit with tall plant, and here capital R is a dominant, so it is a red fruit, and here the both are there having a small t only, that red fruit with dwarf plant. So by the crossing of these two parents will be get into the two offsprings. The two offsprings are one is a red tall plant and red dwarf plant. So in these are the offsprings based upon the options. We have to choose the correct answer. So now option A, 25% will be tall with red fruit, 50% will be tall with red fruit, 75% will be tall with red fruit. All of the offsprings will be tall with red fruit. See now here, we are taken into the two parents. The two parents are crossed, will be taken into the two offsprings. The two offsprings are one is tall red fruit and other one is dwarf red fruit. So the options are they given as a two offsprings. One is a red fruit with tall and other one is red fruit with dwarf. So in the all options are they given as a red fruit only, and here all of the options are they given as a red fruit and all of the options are they given as a tall plant, and the percentage of the plants will be known different. Here out of the two, one of the offspring is a red and other one of the offspring is a red dwarf. So now they're given as options in a red and tall only. So now will be that as a 50% of the offsprings will be going to present in the form of a red and tall, and remaining 50% of the offsprings will be going to present in the form of red and dwarf. So answer is D.